So next we move on to Dr. Allison Reiss. She is an associate professor of medicine at NYU Long Island School of Medicine. She's also the director of the Inflammation Laboratory at NYU Winthrop Hospital's Biomedical Research Institute. Her work has helped elucidate the role of cholesterol flux in the pathogenesis of AD. And more recently, she's been working on the use of human co-culture model of Alzheimer's disease as a biomarker, as well as a predictor of treatment responses, with funding from the Alzheimer's Foundation of America. The title of her talk is a Human Cell-Based Model for Designing Alzheimer's Disease Treatments Beyond Antibody Therapies. We look forward to her talk.
Hi, can you hear me? Yes, okay, great. Thank you so much for inviting me to give this talk. I'm so excited to speak to this group and to hopefully be a catalyst to get us all to do things together, because I think the kind of move forward that we hope to make really requires a huge amount of heavy lifting that is not just going to be here on Long Island. So I'm very excited to get to share with you the kinds of things that we envision. So I want to start with just a little background. So two papers that we published very recently, and Dr. Wisniewski is one of the authors on one of these papers. And the theme here is a lot of the same idea, which is all of the human work where we try to get rid of the amyloid, get rid of the tau. It works very well in the murine models, but when we try to do it in the people, the trials are failing. And like I can give you the latest, I'm looking up what are the latest headlines, what's going on. And we see aducanumab, this human monoclonal antibody against the aggregated forms of amyloid. The panel that's going to look in June in the FDA is supposed to give a decision, but the studies that are done look pretty unimpressive. So we'll see what happens. But I certainly wouldn't be optimistic that this is going to be a real breakthrough in treating Alzheimer's disease. Merck has kind of given up the ghost here, and Lilly and donanemab is another one that is, they keep trying to change the analysis, trying to do things with the data, trying to get something that will, I guess, keeps a huge investment going. But, that's, I guess, not what I think we need to be doing. We need to be breaking out of this, learning lessons from it, taking the results of all these studies, going back and taking all the incredible minds and all the incredible people that are working on Alzheimer's and say, okay, well, it's not working. What are we going to do with that information? So just getting back to fundamentals, which is where I kind of try to go, I think we all see the end pathology of Alzheimer's, the neurofibrillary tangles, the accumulation of tau, the accumulation of amyloid, and the terrible consequences in not just, the shrinkage of the brain and all we can see on all our imaging, but what's left of people, how it takes our souls and our internal memories and all that makes us ourselves and just takes it away. And so that pathology is, I think, an end stage of a process that we know has been going on for a long time. And I think one way to look at it is to say that somewhere in the life of the neurons of the brain was a divergence from the normal to a path of destruction that then snowballs out of control. And I think, looking at it that way, well, maybe we can get in there and stop that from happening. So many diseases where we're making progress thinking in that way. And I like to apply it to Alzheimer's. And no one can deny the importance of amyloid. I think we probably have seen these kinds of slides a million times. Are we going to go down an amyloidogenic pathway or a non-amyloidogenic pathway? And the consequences of which direction. We know some of what's involved. We're learning so much from the mouse models. We've learned so much from all of the genomics and proteomics. We really are learning. And I think we need to reach the bench to bedside with our work. And that's where those families are waiting for us to do that. The mechanisms, we have, like, again, a lot of good ideas, a lot of good information, a lot of possibilities that need to be synthesized in a way that we could take action, that we could do something tangible. And the approaches that we have right now, again, so inadequate. All we have are things that will give you modest symptomatic relief for between 6 and 18 months. In theory, if you could remove or interfere with the aggregated A beta, you're going to improve the signs and symptoms. You can also look at the acetylcholine neurotransmitter, and which is so important in forming and retrieving memories. And if you can inhibit the breakdown, you could kind of squeeze more out of the brain, maybe for a little bit longer, but again, ultimately not changing the history, the natural history of the disease. So the problems, again, the mouse works and then the human doesn't. The heavy investment in this that I think, kind of now throwing some good money after bad. We're getting better at predicting that. I think we're seeing leaps and bounds and tests that are really good, that are blood tests. But if we tell people, well, you're kind of destined to have Alzheimer's, what are we doing? What kind of a grim piece of news is that if we can't help them? So we need to do this prediction, and then we need to offer something, offer some help. And how are we going to do that? How are we going to do it quickly and efficiently? And one of the problems with that is the long time that it takes to see results. I'm not getting any younger, so if I want to see Alzheimer's patients doing better in 10 to 15 years or whatever, I better hurry. And we need ways to accelerate it that, aren't a mouse. Obviously, the mouse has the short lifespan, and that's very helpful. One of our big problems that we don't see in cancer and some other diseases is, we can't go around biopsying people's brains. We can't get those neurons very easily. And if we do get them, they're post-mortem. So we don't have access to that. We need that. I think also we've all seen how when we get to the end stage, by the time people have symptoms and they're compromised, it's really late. The brain is an amazing organ. It compromises so well for so long, but in the end, we're past the point where we really have the chance to do a useful intervention, and maybe we don't show benefit because our damage has occurred. And I think that amyloid and tau are a final common pathway of a bunch of problems that preceded them, and that we have to attack that. And the multiple risk factors and the complexity of the disease somewhat supports that viewpoint. So how are we going to study this? And we're going to look, how are we going to test it? How are we going to do our treatments? That's been kind of the fodder for my brain that our lab has been working on, along with Thomas and with others who are, also trying to make a breakthrough here. And we want to model the human brain the best that we can in a way that we could do this work and kind of get a preview of what's happening in people and do it quickly and efficiently. So one of the great things that's happened in the last few years is the ability of these human neural progenitor cells, the HNPCs from Alzheimer's patients, that have become commercially available. And these are neurons from adult stem cells. So this is like our proxy for the brain biopsy. And they really do behave like neurons from the brain. And we're able to get the ApoE4, the presenilin-1, and non-AD controls, and we have them and we look at them in our lab. And the kind of fun thing to see, or just intriguing thing to see, is all we did here was just grow them on our dishes in our lab and take some photos. And immediately at the early stages, which are A and B, and then the later stages of growth, which are C and D, morphologically, in just gross appearance, the Alzheimer's cells are less robust. And so we were like stricken just by that, just without any manipulation. So the next thing we did was we did start to take a look at some genes that we thought would be important in, Alzheimer's disease and in neuronal health. And we found that synaptophysin, which is important in nerve communication, was decreased in the AD neurons versus the control HNPCs, and LRP was also decreased, which is binds amyloid. So we start to see just at a baseline some differences in these cells. We're also interested in looking at FOXG1, which is a transcriptional factor that's involved in cortical neurons and in neurogenesis and axonal growth. And we see a very big difference in FOXG1. And a lot of this data, is just kind of coming out, or we're in the middle, and we did get interrupted by COVID. We were not able to do a lot of work for a while because of mission critical, and because also our elderly patients that we enroll in our studies were not able to come in. But we're starting to really gear up and get patient samples again. So I'm happy to do that. It was painful to have that time lapse. So one of the key things that keeps every cell alive is its respiration and its bioenergetics. And we know mitochondria are the powerhouse of the cell, and the conversion of glucose into energy is so important. So back to the theory of where this is starting is the interest in HNPC and in neuron behavior in response to glucose, and how that may differ in normal and Alzheimer's. And I think that this bioenergetic difference is going to be very telling in how the cells ultimately end up so sick when you have Alzheimer's. And what is happening in those mitochondria? How might that be entangled in the tau and amyloid formation and the structure and function later on? So this, we're starting again back at that idea of Alzheimer's being like type 3 diabetes. We did, again, very kind of simple experiments, this getting to know these cells. And what we found is that the ADHNPCs, those AD Alzheimer's neurons, when you expose them to high glucose, they increase the base level, that enzyme that is amyloidogenic, much more than the healthy control HNPC from non-Alzheimer patients. So the response to glucose is different. We also found that the high glucose increased APP to a much greater level in those Alzheimer's cells than in the control cells. So glucose metabolism is altered in these neurons. We want to build a close approximation, as close as we can, of an approximation to what the brain does, and to simulate that interaction of neurons with other cells, and most especially the microglia, because they are the immune cells. And we think immunity and inflammation is very important. My perspective is very much of a lipid background and an immune inflammatory background. And so that informs the way that I guess I visualize this system. So we want to take human neurons and the brain support cells and the amyloid producing system from our patients, which is where our personalized medicine comes in. You know, as I mentioned, there have to be patients here. There has to be real Alzheimer's from people in our studies. And that's where we're taking two approaches to do that, to give us the best possible reproduction and replication of kind of what we are able to do with cancer when you can look at personalized medicine in cancer. So as you know, the neurons aren't alone. There's microglia and inflammation in Alzheimer's, and those immune cells coexist with the neurons. And when they are injured, they become like these reactive microglia, and they can contribute to damage and perpetuate a cycle of negativity in the brain. And we know that amyloid activates the microglia. So we want to model the neurons and the microglia together because we think that's a better fidelity to the real thing. So some of the things I can tell you right away from starting this work, and we're right in the middle of doing this, like we have things going on right now. So I hope I'll have more to tell as this story unfolds. But we can tell you that we see some of this cell-to-cell communication again, very simple. We took microglia, we gave them abnormal glucose or insulin, and then we showed that any way that you disrupt glucose and insulin, high glucose, low glucose, high insulin, you up the APP level. And this is kind of what happens in people who really have diabetes. If, any of you who've seen patients, 1 minute their sugar is high, then they're taking their insulin, their sugar is low, then the insulin is low, and we give them insulin and it's high. So I think that the bouncing around, very technical medical term, but the bouncing around of sugar and insulin levels, which is not physiologic, that comes with diabetes, is very damaging to neurons. So we want to personalize the system. One of the ways is using platelets. And the reason is that platelets are really easy to get. You can take them right out of blood plasma. They have abundant amyloid precursor protein and all of the same machinery created by your own DNA as your brain. The platelets are one of those few cells outside the brain that are very actively involved in amyloid production, but it's a lot easier to get your platelets than to get your neurons. And we think that they can be useful in working in our system. And so if your abnormal A beta machinery is found in your neurons, it should also be reflected in your platelets. And again, we get it from that platelet-rich plasma, which they use for athletes to help with joint problems. The Kardashians are big fans for their facials, but platelet-rich plasma is very easy to get out of the blood, just centrifugation. And what we can do is take these platelets from the platelet-rich plasma and take it non-invasively, add it to the neurons of our AD or healthy controls with microglia, and we can measure the effects on all the amyloid machinery and the cell health and viability. And we can then try treatments out. So that's like kind of our first stage of what we're building here. And we are starting, we've done some of this kind of work. We get the platelet-rich and poor plasma from Alzheimer's and healthy controls, and then we can add it to the neurons that we have from the HNPCs that are ApoE4 homozygous Alzheimer's or presenilin or healthy control. And we can learn how the platelet-rich plasma from the AD patients affects the cells compared to the controls. And we cannot just do it in the general sense of AD versus healthy, but specific to the person from whom we took the platelet-rich plasma. And we can follow it to predict the risk in AD risk, and we can evaluate our therapies on our system to see how it changes the amyloid production and the genes of relevance and correlate it to imaging and to other disorders like diabetes. So the first thing we did, and we've repeated this a number of times, is just show that it is the platelet-rich plasma and not the platelet-poor plasma that affects the genes like ADAM10 and BASE. And we did show that this is very much the case. The platelet-rich plasma, when we add it to neurons, affects ADAM10 and BASE, whereas the platelet-poor plasma is pretty much not changing anything or moving any functions. Then we looked at the amyloidogenic properties of the Alzheimer's platelet-rich plasma. So we took human neurons and we incubated them in 10% platelet-rich or poor plasma with AD platelet-rich plasma reducing ADAM10 and increasing BASE, and platelet-poor plasma had no effect. So this is very exciting kind of as a start, again, to what we want to see. And so we see that, again, you're going to get a reduction in the non-amyloidogenic pathway production with a decrease in ADAM10, and you're going to upregulate BASE with the platelet-rich plasma from the Alzheimer's, not with the platelet-poor plasma. Then the other thing we want to look at besides the plasma are the exosomes. So another wonderful thing that we could do as a proxy for getting neurons is your neurons shed these vesicles. The vesicles that come off of our neurons can cross the blood-brain barrier. They have proteins, they have mRNA, they have microRNAs. They're a mechanism of communication in the nervous system. You have bidirectional signaling between neurons and glia and all the cell types that are in the brain. And the microRNAs in exosomes transfer between neurons and microglia and influence their phenotype. That's been shown. And they can affect genes involved in A beta generation. So we can isolate those neuron-specific exosomes. And what we were able to do, again, these are like all the studies that we're just getting started here. We looked at APP, base, and acetylcholine esterase, mRNA, and they are increased in neurons exposed to AD subject exosomes. So we see that the Alzheimer's exosomes versus the control exosomes increase APP, base, and ACHE. So again, showing that, that there are messages leaking from the brain that we can read that tell us when things are going wrong. And of course, it's, you know, it's important to know what's wrong if you want to fix it. And we need to be figuring out ways to do that. So how are we going to use this information? So that's where, again, we see this incredible collaboration to be able to look at this. And Thomas and I have had several meetings and kind of starting to plan, you know, how we're going to do this. And we're very fortunate to have had generous funding from the Alzheimer's Foundation. I'll mention that again at the end, and a further recent infusion of funds. So what we want to look at is the difference in repertoire of the microRNAs in the neuron-derived exosomes from the Alzheimer's and non-Alzheimer's human subjects. And we have an IRB-approved protocol to do that, and we are collecting samples from our patients. We are so grateful to our patients and to our healthy controls, because any study like this, you have to have the healthy people, the older people who have incredible cognitive capability to make those comparisons. We want to do a pathway analysis, look at which of these miRNAs are different in the AD and the healthy, look at what genes they affect, manipulate our system, and see if we can repair or attenuate damage to the neurons in the microglia. So we're building our model to test these exosomes in our system and manipulate the microRNAs. And we want to look at mitochondria and energy usage, because I think a lot of things are going to come back to that. And we want to look at, recently we got the ability to do this and the funding to do this, look at the families, look at the offspring before the symptoms appear, look earlier in the disease where it's in families, and also take away some background noise by having families where you have a lot of other similarity in the genome and the proteome that maybe you can cut down on the noise ratios. So our goal is to get treatments that help humans. Our goal is to make that leap that we haven't been able to make. The precision medicine approach that's used for cancer. We need an in vitro human system to test the treatments. We're trying to develop this system. Advantage here, we're going to use all human components. It's non-invasive. We can use our system to test drugs that hold promise, and we can use the Alzheimer's neurons and the personalized neuron-derived exosomes and the microglia, and then try our putative therapy. We're trying to, again, start on this pathway analysis like it's being done with cancer and with Thomas Wisniewski and the NYU Alzheimer's Disease Center. This is, again, where we see incredible need to collaborate and need to get more funding to be able to bring in more patients, because this needs bigger than, you know, what we can do in our institution, even though we're getting a good start here. The next thing we want to do is start to try to test some things. And I hope there's going to be new things in the pipeline. But some of the examples are things like nilotinib, which is a leukemia drug that's being repurposed and is being looked at to treat Alzheimer's. It's a tyrosine kinase inhibitor. So we're starting to look at, you know, what might that do? If it does help in Alzheimer's, how does it help? And if it does help and we see how it helps, can we refine it or bring in a new drug that's even better? It penetrates the brain, which is a good thing. So we know that it might actually be working. Litaglutide is another one of these drugs that's being looked at. And these studies are being initiated in the National Institute on Aging and in the Imperial College London, respectively. So we're looking at things that are actively being used in people off label. One thing that we found that was pretty interesting was that nilotinib increased ADAM10 message in the presence of our platelet-rich plasma, but not in the presence of healthy plasma. So, you know, there's, again, a little indication that maybe nilotinib has some good effects on ADAM10 levels. We also have just shown in the background with just adding nilotinib to our neurons that it increases in, you know, a dose response manner, in, you know, increases atom 10. And we back up our work by doing a similar study on the effect on base and showing that nilotinib decreases base, not just the message, but the protein. And we're doing a lot of the protein work right now. Heather Renna did a lot of our PCRs, and Saba Ahmed is doing the blots. And, you know, we're trying to get everything to come together before we send it off to you for all that deep dive work. So currently the study that we're doing now is with my colleagues and my clinicians here, Dr. Aaron Pinkasov and Dr. Irving Gomelin. We're looking at males and females age 65 and over. We're doing our screening and neurologic history and examination and activities of daily living and a mini COG assessment. We're excluding them if they have another diagnosis, and we're getting our Alzheimer's patients. We're also looking at our over age 65 controls who are cognitively normal, and we're doing our neurologic history and doing, you know, recent blood work as we enroll these patients. Now, thanks, as I mentioned, to Alzheimer's funding, we're adding not only a greater N, but new groups to our study. We want the first degree relatives of our subjects, that's the siblings and the children over age 18. We're trying to get the people in those early stages to see what happens first. What is the sequence of events so we could get to the early events? So we want those MCI patients who still have a lot of function. So the siblings and the children have the shared genomic and proteomic background, and they can inform by eliminating that background noise that comes with the unrelated subjects. And as I said, we're now funded to increase the N to 50. And we're going to do that analysis of the exosome cargo and the proteomics and transcriptomics with Thomas Wisniewski, our co-investigator there in Manhattan. So what's next? Well, I hope exciting things are next. We need targeted dementia prevention initiatives. We need to analyze the exosomes from the AD patients and the age-matched controls at the NYU Langone core. We need to give our nilotinib studies more depth. We're working very hard on that to look at the mitochondria and the oxidative metabolism in our Alzheimer's disease neurons with nilotinib. And you know, I want you to know how to find me. So this is my contact information. And you know, in the meantime, what do we tell patients? What do we tell people who are worried about Alzheimer's? You know, the age-old things that keep people healthy, keep people with Alzheimer's healthy. That diet, the quality diet, you know, not the refined carbs. Eat well, get physical activity, keep up your social connections, intellectual activity, avoid head trauma, and take care of all your heart disease risk. I give, you know, a lot of talks to patients and families and caregivers. And, you know, we can't underestimate the value of just taking care of ourselves, having the best material to work with if we are going to face Alzheimer's, so that we're ready to do something and have people in good shape to receive our treatments. I have so many thank yous. None of this is possible without all of these incredible people. As I mentioned, Dr. Pinkasov, Dr. DeLeon, and Dr. Gomelin, my clinicians at NYU Langone, Long Island, Thomas Wisniewski, my wonderful collaborator in Manhattan, who is going to do so much of our deep dive work, Dr. Laura Castleman, who does our statistics, Heather Renna and Saba Ahmed, who are doing all of that technical work that you're seeing, Mark Stecker, who really got me started on this path because he came in to do neurology and brought me back to neurology after I had spent a lot of years on cardiovascular disease. It's been an interesting course and direction change in my life. And Irina Voloshyna, my wonderful friend and colleague who worked with me here in Mineola and now is down in Manhattan. And I'm so grateful for the Alzheimer's Foundation. They are endlessly supportive and want this work done and have so much faith in us and in our team. The president and CEO, Charles Fuschillo, the founder, Bert Brodsky, the senior VP, Josie DiChiara, and just so many wonderful people there who you know, work every day to help people with Alzheimer's and who do it in every aspect. I also want to thank Donna De Levante Raphael, who also is always involved in getting our word out to the community. And again, donors, so many generous donors who want to help us. Mr. Robert Busha, the Herb and Evelyn Abrams Family Amyloid Research Fund, Mr. Edmonds Bafford, and Ms. Lynn Drucker. Again, this doesn't happen without people to fund it, because every step of the way is expensive, and I hope worth every penny. So thank you all for your attention.
So thank you for a very stimulating talk. Lots of exciting ideas being put forward. So we have time for a question or two.
There's a question in the Q&A box that somebody had asked anonymously. It says, how well do the HNPCs recapitulate the natural cell type, neuronal diversity in the brain? Even if microcircuitry cannot be fully recreated in a dish, do you maintain populations of GABAergic or neuromodulatory neuron types from which you can then study differences in metabolism?
Well, we can do that to some extent. I mean, it's about as good as we could get up to the minute right now. And I'm always watching for newer and better ways to do it. But this is pretty much state of the art. We're recapitulating neurons of the brain.
I might jump in with a quick question as well, because I think I missed this, Alison, and I apologize. I was interested in how you're studying the microglia. Are you doing this taking postmortem tissue, or are you culturing them? And if you are culturing them, what methods are you using? And I know microglia are very tricky to work with in a dish because they respond to everything. So I was just intrigued.
That is true. So we do our best to grow them. Heather is magical with cell culture. So we grow, we do purchase the microglia and we grow them in our dishes, and we do conditioned media a lot of times to keep things separate. But our plan is to do them co-cultured, at least with, with some membrane separation. So we're just doing, one step after another. But it is true, they are very reactive. But there is, there aren't many alternatives. Everything is just done right now in cell culture.
Yeah, we might have a little chat offline, because I know there's a lot of difficulty sometimes with these purchase lines. They get expanded in serum and maintained in serum, and that causes them to be reactive. And there's a few methods around to get over this. And particularly, I think it would be worth validating the cells that you get sent are really microglia. And there's been some real hiccups in the glial field about cells being sent around that are not what they say on the packet. So yeah, let's have that discussion offline.
Yeah, no, that's very interesting. I mean, for some of the earlier studies, we just use THP1 because as an atherosclerosis person, like I have used those to death, and they are very, very reliable. But I think, we're trying our best. Again, you can use only what is available. And this is again, what we have. And I think that a lot of companies are working on this, are aware that we need to do this as best we can.
Mm-hmm. Okay, thank you.
Okay, stimulating discussion. So I think we'll go on to the last speaker, which is